See, my watch has 10 after 8. My watch has.
Come in late, if you come in after 8, is that back door open? Come in through the back door, don't walk through here. After I started my lecture, okay? Because it's a distraction, students may miss something if you come in late. Okay, with chapter 1, major themes of anatomy and physiology. Now, in this chapter, there's going to be a lot of history covered in this chapter. Your exam has already been made out. You'll have study hints on Blackboard by tomorrow. I think there are 45 questions on it. So we're going to start off talking about anatomy. You got to know the definition of anatomy. I mean, of course, if you're in an anatomy and physiology course, quite naturally, you got to know what anatomy stands for, right? So, of course, anatomy actually occurred before physiology. Anatomy, we're going to look at structures and forms. That's what anatomy is. Physiology, on the other hand, looks at function. You can't have one without the other. So pretty much starting off in 201, we look at a lot of structures or forms, okay? Bones, the skull, organs, those are structures, function, forms, or whatever you want to call them, or organs. But in physiology, that's when you're going to have to translate or transfer the information from here and be able to connect it when you get to 202, which is a lot of functions. I do talk about functions. The last chapter in here, which is chapter 11, we talk about muscle physiology, which students hate. Okay. So I'm going to go, some of this information I'm just going to bypass. Have any of you started your assignments? Have you done any? Now, you know that Learn Smart, there's a deadline for it. That's a quiz. If you don't do that Learn Smart, guys, you're going to have a 0. Remember the other day when I went over the course syllabus with you guys, no late assignments. So if you don't get out there and do your Learn Smart, that drill, I mean, you're not going to get credit for that. It's just something to help you out, to get you through, to help you understand the material. Did you find out that if you edited the trial and then you can still keep drinking? What? And if you get the trial first while you're waiting for the next and you then finally get it for the access to the transportation. Yeah. Okay, so I can start the trial. Yeah. Yeah. You can start that trial, but like I said, I think some students, I was told by another instructor, she says, make sure you tell your students that they can't try and use those trials over and over again, because once you go into Connect, what's going to happen is, with that trial, it's going to recognize you. So if you try and use it again with a different name or a different email address, it's going to recognize you because your information is already stored. So then it's going to tell you, okay, you need to buy this card for $65. Okay? So you guys need to get on the bandwagon, and because I think there's an end date for that quiz for Learn Smart, the little part where you see the brain, the head. I think it's next Thursday or something, because I changed it. So yeah. The last time excuse me? The last time I was okay, well, right now, everything is on Blackboard. I've got to get started with my lecture because if you weren't here the first day, guys, you're going to have to get on Blackboard, read the syllabus, because I'm not going back over any of that information. Everything or you can come by my office during office hours if you have questions, but I've got to get started with chapter 1. Okay, it's very important that you guys come to class on the first day, because what I mentioned on the first day is very important, it's very crucial. But of course, on day 2, I have to start with my lecture. Okay, so everything is on Blackboard. The syllabus, One thing I will say that is on Blackboard, which is a pet peeve of mine, is students coming to class late, okay? So make sure you read that syllabus, and it talks about lateness, it talks about tardiness, because if you come in here on exam day, 15 or 20 minutes after the hour, you will have to take a really, really hard makeup exam. That's in my course syllabus, all right? Okay, now, no more questions unless it's pertaining to chapter 1. Okay, so, okay, there's a definition of anatomy at the very top. It's a study of form. I like to say it's a study of structures, a study of organs, anything of that sort. Examining structure of the human body. There are various ways you can actually examine the human body. You can inspect, you can palpate, you can use a process called auscultation, and you can also use well, sometimes I write. I really don't like to use that because I like to write.
Yeah, I was
Just a little bit. I don't. Like that. I don't get to come right here. You may want to move closer. You mean like right here? Sure, please. Okay, let's settle down because I need to talk. Correct the flow now. Okay. Okay. Now that string is in the way. I don't have time removing that string. So, as I was stating, there are 4 types of examining the body. You have inspection, you have palpation, and of course you have auscultation. Of course, those are in your textbook. Put a picture there, right? On page 2 of your textbook. Okay. When you have an inspection is really just a physical examination. You may go to your doctor, your general practitioner, and they may just do an examination of your entire body. Okay. Palpation is a little bit different. That's where you're going to feel, you're going to touch. For example, ladies, once a month, you know, if you do your self-breast examination, of course, that's an example of palpation, to feel or to touch. Feeling of lumps, tender spots you may have on your body. Of course, that's an examination of an example of what? Palpation. Auscultation, on the other hand, simply means you're going to listen. Okay? Doctors often, like for example, the beating of your heart. Doctors will use a what? A stethoscope, which is their main device when they want to listen to something. The beating of your heart. They can listen for your sounds of your lungs to see if there are any abnormalities there. Percussion simply means you're going to tap. In Anatomy 202, you use a reflex hammer. In Anatomy 202, which actually will test your reflex. Okay? So, a couple of these terms are on your test. Okay? Percussion, the examiner taps on the body, feels for abnormal resistance, and listens to the emitted sound for signs of what? Abnormalities such as pockets of fluid or air in your lungs. In order to really understand anatomy, you have to do a cadaver dissection. A student on yesterday asked if we dissect the cadavers. I said, we don't have the proper ventilation, we don't have enough space to store cadavers. But with your Connect card, there is a cadaver dissection DVD. That's why I keep stressing that Connect card, because oftentimes, like in the past when we used this textbook, I would take that DVD, project it on the board, peel away the layers with a little dissecting blade, and of course, that will be like a muscle exam. All right. I will put numbers up here. So all of that now is a part of that Connect card. You don't have an actual DVD anymore. But anyway, cadaver dissection means to cut up. Dissect means to cut up or to separate, because you want to reveal tissues, organs, structures, and things of that sort. And of course, look at their the relationship that one structure has to the other. Comparative anatomy. Comparative anatomy simply means you're going to take several different organisms or species, and of course you're going to compare their internal anatomy or their structures to view any similarities or differences between the two. Of course you can, well, when I was in college I took a comparative anatomy course, and we would use like mice, sharks, cats, a lot of different organisms, and we would actually compare them. I don't know if they offer comparative anatomy in 2013. That was a long time ago. But anyway, there is a course you can take. Different types of, these are under anatomy, the study of form. These are different ways you can actually examine the body. You can use exploratory surgery, where you actually open the body up. Exploratory surgery, of course, has pretty much been done away with because there are a lot of risk involved in exploratory surgery. And now they're using a lot of what? The medical imaging, like the sonography, the MRIs, the CAT scans, and things of that sort. Okay? Medical imaging, viewing the inside of the body without surgery. Radiology is a branch of medicine concerned with what? Imaging. Gross anatomy, on the other hand, which is one of the subdisciplines of anatomy, of course, you can take a gross anatomy course. I've taken a gross anatomy course. When you take gross anatomy, that's when you look at the cadaver. And you don't need a microscope, because, I mean, you're looking at the human body. You can open it up, cut it open, and of course it will reveal all of the organs and structures to you that can be seen with what? The naked eye. Gross anatomy, of course, is often referred to as macroscopic anatomy. Macro means what? Large. Because those structures are large enough to be seen what? With the naked eye. Psychology, on the other hand, the study of structures and functions of what? Cells, which is also a branch of anatomy. This right here, this area, you know, students come to Lawson and they want to go to nursing school. And this is a hot field right here. Psychologists make a lot of money doing that lesson. Okay? Study, it may be a little boring because you're behind a microscope a lot. The study of structures and functions of what? Cells. When you get a pap smear, okay, they scrape the little cells from your what? Your uterus, and they send them to a lab. Then, of course, psychologists will actually what? Examine those cells to see if there are any what? Abnormalities there, or if you've got any type of abnormal growth or something, or whatever. Okay? If you go on down, these terms, a couple of these, I know psychology is on your exam. I normally pick out the major terms and things that I think are very, very important at this point in anatomy. Histology in the form of microscopic anatomy. Why do you think this one is microscopic anatomy and this one is macroscopic anatomy? You have to use a microscope, exactly. Examination of cells, actually tissue cells with what? A microscope. In chapter 1, it mentions a lot of the terms in anatomy are Greek and Latin origin, which means they can be a little bit difficult to pronounce and a little bit difficult to actually spell. But if you understand, it's so important to take medical terminology, because you're going to understand. If you understand the prefix, the root, the suffix, of course you will be able to put these terms together without even looking in a medical dictionary of some sort. A couple of these will be in the back of your textbook. I don't know if any of you have it yet. Who has your book? You have at the very back what you call a lexicon. A lexicon, of course, is just biomedical what? Word elements. Okay? So, if you break down psychology, who has a textbook? First, if you have a textbook, write this word down for me. Cyto, okay, which is your root. Find out what cyto means. I'm sure everybody knows what logy means, right? Cyto means cells, right? And logy means what? Cellular white cells. What about histo? Histo means what? Who has the textbook? It means tissues, right? Okay. Study of what? Tissues. We got examination of cells. What they have in their textbook on page, should be tissues. Histology. To see those, we usually take tissue specimens, thinly slice and stain them and observe them under what? The microscope. So guys, histo means what? Examination of cells or tissues. We're going to say tissue with what? Microscope. Neuropathology. Neuro means what? Path. Now here you've got several roots going on here. Pathology. What does neuro mean? It's your brain, right? Patho means what? Disease. Logy means yeah. So you're going to examine the brain or the study of the brain for any type of what? Disease. Okay. And you also have neurophysiology. You got neuropathology, neurophysiology. So is neurophysiology. You gotta look at the function of what the brain, right? Exactly. Good. Okay. So with ultrastructure, there's another one right here. Histopathology. Okay? So you're going to look at tissues for what? Study tissues for the signs of what diseases, right? Ultrastructure, you guys, well, in the biology 103, we talk about revealing cells using the electron microscope, the transmission, as well as what? The skin and electron microscope to get a detailed examination of cells. All right? Okay. You guys with me? And those of you that are here for the first time, I'm in Blackboard. These slides are on Blackboard. You can bring your laptops, your iPads, or whatever you have in here and pull up these slides, or you can just actually print them. Okay, these are your learning outcomes. Let me just skip right over that. Okay, now we're about to get into a little bit of history of anatomy, which is kind of like the fun part. Of course, you can see that medicine started about 3,000 years ago, 4,000 years ago. During that time, they didn't have a lot of the stuff that we have today in medicine. They were using herbal drugs, salts, and physical therapy because they believed that really the body could heal. You could heal your body by using things such as that, which of course I believe will hold to be true as well. A lot of the physicians back then, as you're reading chapter 1, they weren't trained. They did not go to medical school. They were performing surgery on people without washing their hands or sterilizing the equipment, spreading a lot of infections throughout the hospital. Pregnant women during that time, a lot of pregnant women were dying. The babies were dying. Well, the mothers too, because they were of the infections. I mean, they would just walk off the street in their regular clothes and just start doing surgery on people without using the proper sterilization techniques. Okay, one of my favorite contributors was Hippocrates. Okay, you're going to see this guy again. He's considered the father of medicine. This was the guy, he established the code of ethics for medical students that's recited to this day. All right. He urged physicians to seek natural causes of disease rather than attributing them to the acts of gods and demons. So he kind of wanted to use a holistic approach to treating patients, and not just them saying, okay, some type of demon or something. So he wanted them to take it a little bit deeper to understand what really is the cause of a lot of diseases that we have, which I think is very important too. Aristotle, okay, one of the first to write about anatomy and physiology. Let me just pull out this. We believe causes of diseases were either supernatural or they were what? Natural. Okay? So there were two terms, okay, came up with theology, theology, or physiology. The terms that we use today, such as physician and physiology, of course, are derivatives of this one is qualified what? Naturally. Okay? We believe that complex structures are built from simpler parts. In your textbook in chapter 1, complex structures, of course, going from simple to what? Complex. You have an atom, you have what? Molecules, you have what? Organelles, you have cells, tissues, organs, and what? Organ systems. So you go from small to what? Largest, or from the simplest to what? The most complex. If you guys took Biology 103, they're called, well, here they're called levels of hierarchy, but I think in Biology 103 they were called levels of organization. You're going from the smallest to what? The largest or the most complex, which, of course, you'll see later. Okay. Claudius Galen, unfortunately, during his time, you're going to see this guy again, he only dissected, cadavers were banned during his time. A lot of unethical practices or things were going on during his time when he was a physician. So the Roman Empire or the powers that be decided to ban cadaver dissection. Students would go to graveyards and dig up bodies and dissect them. So that's when it became, they decided to ban it. So he dissected about 2 or 3. That's when, and then after that they banned it. So most of his work he did work with animals, like pigs, monkeys, baboons, things of that sort. And a lot of his findings were incorrect because he did not do dissection on humans, but on animals. The structures were a little bit different because, like the lungs, if you look at the lungs over there, you can see that lungs have 4 lobes. He said that lungs have 5 lobes because in a baboon, of course, you're going to see, along with 5 lobes instead of 4. So then that's when he decided to write this book. And he told others that came behind him to trust their own observation and not what's written in his textbook. A lot of people ignored it, so a lot of incorrect information occurred during his time because they were reading the works of Galen and Aristotle. And of course, this went on for thousands and thousands of years, which was pretty unfortunate. And then you had the birth of what? Modern medicine, the rebirth of what? modern medicine. Okay, this is my boy right here. I don't know if you can see him, but Andreas Vesalius, he's on page 4 and the top of page 5. Okay, now this guy was an anatomist. Okay, this is where science was reborn. When this guy came into the picture, he taught anatomy in Italy. During his time, that's when cadaver dissection was brought back into the classroom. They could perform autopsies on people that had died, such as slaves, prisoners, people of that sort. The students could actually dissect them. So they allowed that. But also during that time, a lot of the anatomists during his time, they wouldn't, because you did not have refrigeration, you did not have embalming, they would have to do dissection outside. So can you imagine doing a cadaver dissection when it's 100 degrees out there? Some of y'all would probably change your majors if I had to take you out there and say, don't come back in until you finish dissecting that cadaver, that dead body that stinks. Because it was not refrigerated, they did not have the embalming, that's why they did it outside. And they would start with the areas of the body where the organs would putrefy the quickest, which was like the abdominal region, your guts, okay? Small intestines, large intestines, the stomach. They would start here, work their way up to the chest cavity, okay, then to the head, the neck, and they would do the arms and legs last. But those anatomies thought they were all that, and then some. They would sit, how does your book describe it? In these high-ranking chairs, okay? They would sit up high, and they would read out of the anatomy textbook. And, of course, they had this poor little barber surgeon here who was at a lower rank. They would have him pull out the organs, and he would hold them up for the students to actually see, okay? So this guy here, he performed his own dissections. He came off the throne and he started doing this dissection with the students themselves. Barber surgeons, barbering and surgery were considered kindred parts of the knife, simply meaning those reddish white stripes that you see. You still see them some throughout Birmingham. Those little barber poles that you see, of course, came from this right here, which means blood and what? Bandages. The red for the blood, the white for what? The bandages. Okay. So, page 5. In those days, before refrigeration or embalming, the odor from the decaying cadaver was unbearable. Dissections were conducted outdoors in a non-stop 4-day race against decay before the thing just rotted away. Okay? Weary medical students had to fight the urge to vomit. Some of you will probably get really, really sick and probably would change your majors. And I would say, just go vomit and come back. Okay? Lest they incur the wrath of an overbearing home professor. Professors typically sat in an elevated chair, the cathedra, reading dryly in Latin from Galen or Aristotle, while a lower-ranking barber surgeon removed the putrefying organs from the cadaver and held them up for the students to what? Ugh. See. Okay, William Harvey. That's my boy, too. Okay? This guy was a physiologist right here. Okay? Contributions represent the birth of experimental physiology. Okay, he was the one that actually discovered. He did a series of experiments, and that's when he was the actual one that described how the blood circulates through the body. The blood circulates through the body and returns back to what? the heart. He and Michael Servetus, okay, his partner in crime. However, during that time, because people believed the works of Galen and Aristotle, the people in the medical community thought they were like wackadoodles or crackpots. They were like, what are you talking about? Blood doesn't circulate through the body and return to the heart. No, that ain't right. But, of course, it was proved. He proved it. The Chinese had already stated it prior to him, but he actually did the experiment to actually prove it. And it took a long time for people to finally, what? Accept it. They ridiculed him for his what? Theory. Okay? So he and Michael, they were the first Western scientists describing how the blood circulates to the body from the heart, and it goes back there to the heart. So I know you guys know who this next person is, Robert Hooke. What do y'all know about this guy? Biology students. Let me take my glasses off so I can see. Don't look at the slide. Who took biology, principles of biology, or just general biology? Did y'all? Well, I know if you had me, we talked about Robert Hooke, Anton van Leeuwenhoek, Slyden and Swan, Leslie, right? Jasmine, Tremaine. Who else was? Kelsey. Who was this guy? What did I tell y'all about this guy? What is my biology class? Well, yeah, he did. Look at the microscope right here. Anton van Leeuwenhoek was the actual guy that invented the microscope, but yeah, he did make improvements with the microscope, like with the ocular and the objective lens. Okay, magnification wasn't that great. What is he really noted for? Which you will probably see on your exam. Listening. What? Yeah. He coined the term what? Cell, right? He was the first to what? To see or identify what? The cell, and he coined the term. That's where the term cell came from. This guy right here, Robert Hooke. This is what he looked at under the microscope. Cork, which is the dead cell wall of what? A plant. Right, Mr. Hill? You were in biology. And you see these little boxes right here? That's what he named them, or called them what? Cells. So he, first to see and name cells, so he coined the term cells, published the first comprehensive book of what? Microscopic. So he, Robert Hooke and Anton van Leeuwenhoek, they took anatomy to the cellular level, per se. Now, who was Anton van Leeuwenhoek? Okay. Was he invented the single lens microscope? Okay, and what else? Guys. What is he really noted for? Because he was really the first one. He wasn't a physician. He was a seamstress. This guy was. But he sold clothes. Okay? I guess he was bored one day and decided to invent. He took a magnifying lens and he started looking at a lot of things in his little place of business. And that's when he invented the microscope from that, because that magnifying lens made everything what? Larger, right? So what is he really noted for? First to look at what? To discover? Exactly. You will see it again. I need to make sure I put that on my test. You observe what? Microorganisms, right?
So what he actually did, he would take, he would scrape his teeth, the bacteria on his teeth, and he would put it under the microscope and he would examine it, and it was moving. So if you scrape bacteria off her teeth, I don't know if it was moving now, but yeah, he saw it moving. You look at pond water. When you look at pond water in biology, you now see some stuff moving. You look at pond water. Y'all were in that fall class, right? Leslie? Tremaine, you were in what? The spring, yeah. I had them collect pond water. So y'all didn't bring any. So yeah, and they had to take like little droplets of pond water, and that's what he did. He took pond water, he took lake water, he took specimens from wounded soldiers, the pus from their wounds, and he would look at it under the microscope and see little microorganisms in the pus. Sperm cells, he would see them swimming, moving, and he called them what? Little what? Animalcules, right? Y'all remember that word? I think it's in your Biology 103 textbook. Okay. And of course, this guy here, these are what? Tenets of the cell theory. With improved microscopes, examination of a wide variety of specimens followed, concluded that all organisms were composed of what? Cells. So you guys remember the cell theory, right? All cells are what? They're living, right? All cells are living. That's one of the components of the cell theory, right? Cells come from what? Pre-existing cells, right? Okay. All living organisms, whether you're a plant or an animal, have what? Cells, right? Okay. So that's the cell theory from biology. Here's Anthony van Leeuwenhoek, invented the simple microscope with great magnification to look at fabrics. He was a Dutchman. Published his observations of blood, lake water, sperm, bacteria from tooth scrapings, and many other what? Things. Here are these guys too. They eliminated the blurry edges of the microscope, so they increased the focusing power, or they added the condenser lens to the microscope. So you guys know about Charles Darwin. Of course, he's the guy that came up with this theory of what? Natural what? Selection, how species originate and change through what? Through time. Of course, we evolve. When evolution occurs, it's when there's a change in your what? Your DNA or your genetic what? Composition. Populations of what? Organisms. So there are selection pressures when you can adapt to changes in your environment, which are AKA known as selection pressures. The survival of the fittest, those are the ones that are going to what? Survive. Those are the ones that are going to what? Repopulate what? The area. Those are the ones that are going to what? Reproduce and create what? More of themselves. Selection pressures like climate, predators protecting themselves from other animals, food, being able to get to food. So there's a lot of competition going on in the environment. And I was talking, I think my Biology 103 students, I was saying when I was a little girl, butterflies were very popular in the springtime. I mean, you could see hundreds of those things. But I think now, butterflies now are considered an endangered species because I don't think you see a lot of them now. Because one of the main reasons is because what we're doing, we're cutting down or getting rid of a lot of these organisms, natural habitats, like cutting down trees, because certain types of butterflies, they migrate in the fall to warmer places. And of course, that's where they kind of like hibernate
They go into their little, they reproduce, make cocoons and whatever else. And then, of course, in the springtime, they what? But there's all those babies, okay? But they gave a couple of ones in your textbook, bacterial resistance to what? Antibiotics. You guys are going to study in microbiology how certain changes in some of their organelles such as their ribosomes, can actually change the cell membrane, the cell wall, can actually change to allow them to to become resistant to a lot of the antibiotics, because they can actually evolve. There's so many different types of strains of AIDS virus out there because of the genetic composition, of course, has evolved the RNA. Same thing with the influenza or the flu virus. There are a lot of different strains of that out there as well because of evolution. As I told you before, some individuals within a species have hereditary advantage over their competitors, which is natural selection, and of course being able to adapt to these selection pressures here allows them to reproduce. Right? Know what selection pressures are. So everything changes about them, their structures, their functions, their behaviors, okay? And then it goes on to talk, okay, they say our cousins is the chimpanzees, according to this book, not mine. Okay, the closest relative to us guys, of course, is the chimpanzee. There's only a 1.6% difference in their DNA and our DNA. So a lot of their anatomy and physiology for their structures and functions are similar to ours. But their DNA is more closely related to ours than the gorilla. Okay? Chimpanzees and gorillas, they differ by 2.3%. Because they're saying we didn't evolve from a monkey, but they're saying that they're our cousins. They say they're y'all's cousins in this book right here. Okay? Study of evolutionary relationships helps us choose animals for biomedical research, which is called the animal model. Rats and mice, of course, they do a lot of, in order to test drugs, they have to try them out first on animals, right? Before they actually give them to humans. So rats and mice, they're easier to order. They're cheaper. They're used more extensively. There are a lot of ethical issues with using chimpanzees. The cost. One of them they're very expensive. So they don't do a lot of experimentation with chimpanzees, although there's only a 1.6% difference in their DNA and our DNA. However, if there were no issues of cost, availability, or ethics, we might test drugs on our close living relatives, the chimpanzees, before approving them for human use. Maybe they can identify a lot of those side effects that comes with those pharmaceutical drugs. If they did research with our cousins, right? Unfortunately, they don't. You guys with me? Let me just vestigial organs. We're going to talk about this piloerector muscle in another chapter. Guys, make sure you always read deeper insight throughout the chapter. It gives you a lot of pertinent or important information. So on page 10, we talk about vestiges of human evolution. But, I mean, you can read about it, but I really don't get off into talking about it. But like I said, we're going to talk about the pilo because, as you can see, these structures or those muscles don't have any purpose in us. So when we talk about the piloerector muscle, when we talk about the skin, that's that muscle that's in your skin when it's really, really cold outside to cause you to have the goosebumps. Or if it's really, really cold in here, that muscle will actually contract and it causes those little hairs on your arms to stand up, and then all of a sudden you get those little bumps. But, I mean, it just happens out of nowhere. It doesn't have a purpose or anything. I mean, they just occur. However, it does have a purpose in certain other organisms. When they're, like bears, when they hibernate, certain animals, when their piloerector muscle contracts, of course, and the hairs stand up, it keeps them warmer. Just like with this auricularis muscle, which is in your ear, doesn't have any purpose for us, but does in certain animals like dogs. When their little ears are moving, they know what you're saying. You do need to know what arboreal is, that second term that you see up here, because the earliest primates, they lived in trees. That's what arboreal means. They lived at the top. If you go to the Amazon or tropical rainforest, you're going to see a lot of organisms at the very tops of tree limbs. That's what arboreal actually means. It means tree top. So those early primates, guys, they were arboreal. They moved to trees due to what? Safety, food supply, and what? Lack of what? Competition. And a lot of their food supply they were eating insects anyway, and a lot of those insects, of course, lived in trees. So some adaptations actually occurred here. If you'll look in your textbook, those adaptations for treetop lifestyle. Okay. On page 11, those adaptations, those were some changes. These are some changes that actually occurred in primates, okay? The mobile shoulders, which is similar to ours, okay? That allowed them to reach, to swing from what? Branch to branch, or what? Limb to what? Limb, mobile shoulder to reach what? Overhead. They have the opposable thumb. So they're showing the opposable thumb. That's when you take your thumb, and of course you can touch your fingertips. Okay, they're showing you chopsticks in your textbook. So you take the thumb, and you can actually touch what? Another finger. They can cross the thumb to touch what? The fingertips. It's called opposition. We're going to talk about thumb opposition when we get to chapter 9. Prehensile grasp allowed them to grasp or hold on to what? Limbs or what? Branches, right? Okay. They had the stereoscopic vision. Their vision improved to allow them to see at a distance for that 3D image. Okay. That perception for leaping and what? Catching what? Prey. Color vision. Okay. They knew they could distinguish the color of fruits, which ones were ripe, which ones were not ripe. Okay, just by looking at their color. Less toxic foliage. The young tender leaves from older, or the leaves that are not so tender. Okay, And of course, they develop larger brains and good memory, just like us. They can remember their food supply, where it came from, what part of the forest or their natural habitat, and they were capable of going back there to actually get it. Remember the food sources and improve social organization. Because they had social media too, like Facebook. Okay. Monkeys, apes, and humans do, however, share common ancestors. Our relationship is not like parent and child, but more like cousins who have the same grandparents. Observations of monkeys and apes provide insight into how primates adapt to the arboreal habitat, and therefore how certain human adaptations probably originated. And then, guess what? They started walking upright. Okay? When the area, African forest, when it became more terrain or more terrestrial, they stood up. Okay, because they had to see over that tall grass, all of those trees, things of that sort, producing more predators and less protection. So a lot of adaptations occurred here. It's called bipedalism, when you're standing upright. Okay, so they went from here on all fours to here, right? Standing and walking on two legs, they could spot their prey when they were actually standing upright. They could carry their food. They could have their weapons ready for attack. Okay, so in order for all of this to occur, those adaptations to occur, which is very common in us now, adaptations occurred with their skeletal muscles, the skeletal features, the bones, which is the femur, the hip bone, or the pelvic area, as well as the muscles. That increase brain volume. Okay. Okay, you guys already know about this, so I'm not going to talk about it. All right. This hierarchy of complexity, going from simple to what? Complex. I think that's one of the questions on that activity that you guys have to do in Connect. When are you guys going to get your Connect card? Who has their Connect card? Okay, only 3 people? Of next week. So you have up until next Thursday to get that quiz done. Guys, I can't keep putting this off, okay? Now, like I said, if you don't get that quiz done by next Thursday, you're going to see a 0 on Blackboard. Now, I don't give the 0. Connect rates it, and it dumps it over into Blackboard. Remember, I told you guys, once you complete that quiz, and you hit submit, you're going to have to click where Return to Blackboard, and it's going to shift your grade over there into Blackboard. So if by any chance, I didn't give you the 0 now, Connect breaks that stuff for you. Connect says, she missed the deadline, or he missed the deadline. A 0 it is. Shift it over to Blackboard. Or if you don't go in there to do it, you can't send it to Blackboard, then I can go into Blackboard. I can also go into Connect, and I can look at each of your individual names, and I can see who did not do it, okay? And, of course, your grade will, and then that's when I go in and record a 0, because I have to do something on my end when you don't do it, or don't send a grade over to Blackboard. So, just letting you guys know, because we are moving right along. You got it? So that Connect is it's really going to help you pass this course. So Connect is your friend, really. Weren't you in my biology lab? Yeah, y'all tore those pages out.
Yeah. No copies. We want copies. Your first lab will be on Blackboard by tomorrow.
Instructions for it. So, did I put you guys in groups? Y'all in groups? So, if you're here for the first time, you got to. When is y'all's lab? Tuesday, right? Next Tuesday? Yeah. So your first lab is on Tuesday. Like I said, now, if you're here for the first time, okay, let me just say this. The first thing you need to do, if you have not, is to read that syllabus, okay? I do not take late assignments. But honey, look, as someone that have taken me before, this is a serious matter here, okay? You come in here, it's very serious. I take my courses very serious, okay? Because that's just how I am, okay? So don't get off into, if you don't want to see my attitude, don't get off into coming up with a lot of excuses as to why you didn't do it, because I really don't want to hear it. And the grade is not going to change. So it is what it is. You know, it's going to be what it's going to be. If you don't do what you're supposed to do in here, it's going to reflect on your grade. Okay? So that's why I keep talking about that connect thing and that quiz. All right? Because it's going to really help you. It really is. That connect, they say it's supposed to be really good. I mean, this is my first time using it with this textbook, so I'm looking forward to having a lot of A's in anatomy and not one or two. Okay. Now, where was I? So, we know that molecules, look at the atom. Smallest particle in both what living and non-living things. Atoms become what? Molecules, right? So molecules are composed of what? Atoms. This looks like a water molecule. The H2O, the oxygen with the two hydrogens attached to it. Okay. And of course, you have macromolecules. You guys remember macromolecules from biology 103. What structure is this? It's the DNA, right? DNA is what? A macromolecule, your proteins, your lipids, your carbohydrates, right? So you have your macromolecules, and of course you have organelles. So organelles are composed of what? Molecules. What organelle is this? Okay. Organelles are found in cells. Now look at the cell right here. Cells are living, right? Cells become what? Tissues. When you have two or more cells, two or more, look at it here. It becomes what? A tissue. You've got 3 cells here. That is what? A tissue. There are the next chapter, chapter 5, which is on Blackboard, the PowerPoint. We're going to talk about the 4 different types of tissues: the epithelial, your connective, your muscle, and your neural tissue. Tissues. When you have 2 or more tissues, it becomes what? An organ. This pouch right here is what? Your stomach. Organs become what? Organ what? Systems. What system is this? Digestive system, exactly. And of course, the highest, all of this leads to what? A multicellular what? Organism. You got it? Or an organism which is composed of organ systems. You have 11 organ systems. I think they're in Atlas A. There are, I think I put, there's several on your exam that's coming up. So, yeah, go on and get the, do what you need to do. What time is it? Okay. What time do y'all leave? 9:15? Okay. Anatomical variation. Yeah, read about that. 70%, no two humans are exactly alike. Okay? There's certain structures that we have that are, like with the lumbar vertebrae, the backbone, the lower back. Typically, I would say a normal person, but we're all moving, right? These are the lumbar vertebrae. Some have 5, some people have 4. Or lumbar vertebrae. Some people have 3. I mean, they're still normal. I mean, but they think, according to anatomists, they base it on 70% of the people that they study that have the most common structures. There are variable number of organs, such as missing muscles, extra vertebrae, renal arteries. Some people may have 1 kidney instead of 2 kidneys. But some people may have 2 spleens. So there's going to be a variation going on there. It mentions the muscle, the palmaris longus, which is the muscle that's found right here in the palm of your hand and goes all the way down here, the tendon does. But I mean, if you don't have it, then there's nothing wrong with you. I mean, you can actually see the tendon here. If you make a fist and kind of flex your wrist a little bit, you can see the tendon here from the palmaris longus muscle. And it's really useless. A lot of times, physicians, they just take the tendon and they use it as a graft in other parts of the body. The popliteal, the plantaris muscle in the textbook, which is back here in this little popliteal region. Some people don't have that. So there are variations. Okay? So they show no two humans are exactly alike on close inspection. Even identical twins exhibit what? Differences. Yet anatomy atlases and textbooks can easily give the impression that everyone's internal anatomy is what? Same. This simply is not true. Books such as this one can teach you only the most common structures, the anatomy seen in about 70% or more of what people. Someone who thinks that all human bodies are the same internally will make a very confused medical student or an incompetent what? Surgeon. Okay? So it's going to be different. Variations in organ location, situs, soletus, situs inversus, dextrocardia, which means your heart is on the opposite side. Situs perversus. Situs inversus simply means that certain structures that should be on the right side are actually now they have been what? Reversed. They're on the left side. Okay? In your textbook, page 15, I'm going to stop here. And I'll talk about that when you guys come back on Tuesday. And we're going to finish up chapter 1 because you can see I only have 4 more pages left. I'm going to talk about positive and negative feedback, which you will definitely see on your test. Okay? You need to understand, and then I'm going right into Atlas A and talk about directional terms and cavities. Okay? So, any questions? So I just recorded this lecture